Hello, everyone. Welcome to The Secret Life of a Hospital. My name is Charlie and I'm the Engagement Support Coordinator for the Faculty of Medical Sciences at Newcastle University. And I just wanted to very briefly jump on and say hello, welcome. Thank you so much for joining today. Thank you for coming to The Secret Life of a Hospital. This is one of the most popular events that we've run in the last year digitally, so I'm so excited to see so many of you in the room today, joining today's webinar, interested in learning more about the clinical research that's been going on within Newcastle. The Secret Life of a Hospital was conceived as a celebration, as a way to mark International Clinical Trials Day. International Clinical Trials Day happens each year in May to celebrate the anniversary of the first clinical trial in 1747 into the causes of scurvy on board the HMS Salisbury. In Newcastle, we're home to a range of brilliant clinical research that's being delivered in the local hospital and in the university facilities. So we wanted to take this opportunity this year to really celebrate some of the brilliant research that's been happening over the last year during COVID-19. We've got some fantastic speakers lined up for today, looking at some really interesting topics, and I'm really excited to get going, as I'm sure a lot of you are. You don't want to be sitting listening to me right now. But before I can hand over to people who are a lot more interesting than me, I do very quickly need to go over the Zoom etiquette. Now, I know what you're thinking, it's been over a year now of doing things digitally. We've all used Zoom before, and I don't really need to be doing this. But just in case, A, no one's actually used Zoom before on this call, which if this is your first time, thank you so much for joining today and letting this be your first Zoom experience. But B, you may have noticed we are not in the normal Zoom, we are in Zoom webinar. So for some of you, this may be the first time you've used Zoom webinar. The biggest difference between Zoom webinar is that you can see and hear us, but we can't see and hear you, which means that you have full permission to get as comfy as you like, be in your pajamas, have a cup of tea, and watch today's speakers in the comfort of your own home completely. But that doesn't mean that you can't engage with us. So there are some buttons along the bottom of your screen. You should see a button that says leave. Don't press that. I'm hoping you're going to stay for the rest of the call. You'll also see a button that says Q&A. This is the most important one. I'm hoping you're going to have questions for our speakers. You might want to probe for a bit more information, and this is the best way to do that. So you can type in questions, you can send them anonymously, or send them with your name to each of our speakers to ask a couple of questions that you want. And the brilliant Dominic is going to filter through some of those questions and ask some of the most popular ones to some of our speakers today. How do we know which ones are the most popular ones? Well, again, that's up to you. If you go in Q&A, you might not want to ask a question, but you can look through and like the ones that you want to see asked, and that tells us which ones are the most popular to ask to some of our speakers today. There's also a chat function, so if you want to send in comments, tell us how brilliant we are. You can also do that, although I will say the chat won't really be looked at as much as the Q&A. So it is the Q&A that you really want to focus on. Also, just to say that today's event is being recorded. Obviously, we can't see and hear you, so that's not really a big deal for you. But it's just so we can make it available online for anyone who wasn't able to come along today. But other than that, I think that's everything that I need to say. It's the beauty of you being in your own home. Is that if you need the loo, you should know where it is. And if there's a fire, do evacuate your own building. You don't have to stay for the whole event. But I think that's everything that I need to say now. So I'm going to stop talking and hand over to people who are a lot more interesting than I am. And we're going to kick off today with Professor Fai Ng, who's going to give us a little bit of an introduction to the NIHR Newcastle Clinical Research Facility. So I'll hand over to you, Fai.
Can everyone hear me okay?
Yes, yeah, Fai, we can hear you.
Yeah, because I saw that somebody saying on the chat box that they can't only just barely hear us. Now, I apologize that unfortunately in my office, I haven't got a video camera, so you won't be able to see me. I know that all the other speaker, you will be able to see them when they are presenting. So first of all, extending my welcome to all of you who are joining this webinar today. And it's a great pleasure for me to tell you something about the NIHR Newcastle Clinical Research Facility, which I recently taken over from my colleague, Professor Mark Walker, who has been the director for the past 15 years, including setting the facility up. So I'm relatively new to this role, but I hope that I can still do the justice in terms of telling you something about the Newcastle Clinical Research Facility, which I probably refer to Newcastle or just NCRF later on in some of the talk. Now, so I'm trying to move it along. So, and also this is also, if any of you think that this is new to you, this is also new to me as well, this webinar. So I hope that there's not too many technical glitches. So what I'm hoping in the next 15 minutes or so is to tell you a little bit about what is the NIHR Newcastle Clinical Research Facility and why it is necessary and what does it mean for the patient and the public. And then finally, just to tell you a little bit about our vision going forward. Now, so the NCRF is, in fact, is not a single location. In fact, we have 3 locations. We have the Clinical Aging and Research Unit in the campus of Aging and Vitality, which was the old General Hospital site, but is now part of the Newcastle University. We also have the Dental Clinical Research Facility in the Dental Hospital, and also the Clinical Research Facility on the top floor of the Lisa's Wing at the RVI, where my office is based. Now, so exactly what is the NCRF then? It is realistically, it is a bespoke facility to support experimental medicine study and early phase clinical trial. Now, I hear you say, what are the experimental medicine study and early phase clinical trial? So effectively, the experimental medicine study are studies that are taking place in human, mostly about finding out mechanism of diseases or to generate some kind of proof of concept data regarding either some new discovery or treatment. So for example, it may be that a treatment for diabetes that we wanting to show that is actually can increase the insulin level. So of course, and you will still need to have more tests and trial to confirm that it's actually work. So that take it on to the early phase clinical trial. So these are typically the first in man or first in patient study. Now, the aim of this study are mostly focusing on safety and tolerability. Now, those two are not exactly the same. So because the drug can be perfectly safe, but it's not necessarily tolerable by the patient. So for example, it may be that you need to take the drug too frequently, or maybe the mode of administration are different, or maybe some of those things are perfectly safe, but it's such a nuisance for the patients. It is also important to understand what the body does to the drug in those kind of early phase studies. Basically, that would mean that how the drug are being absorbed, being distributed, in different parts of the body, and how the drug are being get rid of. But of course, it is also important to collect some preliminary data about whether this drug or this treatment actually potentially work, but it's never going to answer the question definitively. That would need further trial later on to prove that. Now, so just to summarize about the journey from discovery to actually, treatment that are being benefiting to the patient. So we often starting off with the laboratory kind of medicine, so mostly things that carry out in the laboratory, including potentially animal study where relevant. It would give us some basic science knowledge that would take us on to the experimental medicine that we're wanting to test it in human. And that would probably help us to understand the disease mechanism more clearly and identify some candidate of treatment or strategy for treatment. That would then be tested in early phase clinical trial. As I said, that is is important to find out whether they're safe and whether they're likely to be effective. And then it will then move on to find out whether they actually, effective. That would be the later phase clinical trial. And if they are successful, then it will be about trying to implement them in the clinical practice. Now, of course, the process is not one way. It is also reverse as well. So we will learn from each step, and it may inform going back into the previous step and advance our medical science and treatment strategy. Now, so the NCRF, therefore, are mainly focusing on supporting this middle part of the journey. So about supporting experimental medicine and early phase clinical trial. So why do we actually need a bespoke facility to support those studies then? So the reason is because early phase clinical trial and experimental medicine study are often very complex. The study designs are complex, and also there is a very large element of unknown, including, you will probably heard this quite a lot now these days, unknown unknowns. So therefore, actually, in this study, often it involved many different tests. So some of them are targeting towards understanding the safety, some of them about the pharmacokinetics, which is what I explained earlier, some of them about finding biomarkers, whether it's in the blood, in the tissue, or imaging, and also about understanding mechanism. So which means that this study often involve very frequent and very close monitoring, and also there will be very strict regulation, compliance, and governance issue. And therefore, to run this study and to support this study, we need an expert both in clinical science, scientific knowledge, as well as the right skill and expertise. And of course, communication between both the patient, the research staff, the investigator, or sometimes we call them sponsor, and the regulatory agency are of critical importance. Now, so basically, for purpose-built research facility, we of course would have the standard kind of area, waiting area, the consultation room, inpatient facility, and of course in the dental cases, the dental facility. But also on top of that, we have a lot of very different bespoke tests. Many of those are very advanced. We can check the gait, we can check different imaging, we can do various neural electrical test, and of course we have a laboratory, we can have an exercise lab, we have a children dedicated area. So a number of different things so that we can run this study effectively. Now, but of course, the NCRF is not just a location, it's not just a building, it's not just facility, but actually also about having a very skilled workforce with the specialist knowledge and experience. So this including our research staff, so whether they are nurses, physiotherapists, clinical trial assistant, and healthcare assistant, research scientists, and doctors. But also the support staff, they are also equally important. The clinical trial coordinator, data manager, finance, administrator, But of course, at the management level, they also need to understand what are the importance. So we have the operation manager, Dominic here, our senior research sister. We have somebody to make sure that quality is good, research matrons and the directors. But also not about people, but also about the actual operation environment. So that would including making sure there's a right level of training, learning, development, and also to assure that the quality that the study that we done are of high quality. Now, but the NCRF really never work alone. So in fact, as you know, the NCRF is actually a partnership of the Newcastle University and the Newcastle Hospital Trust. But even the two partners itself is not enough. We actually work very closely with the Newcastle Biomedical Research Centre and also a number of various other organizations, both locally, regionally, and nationally. But perhaps most importantly, I should say, it is actually you that we need to partnership with. So, and patient and the public are really at the center of the partnership, because ultimately what we're wanting to do, hopefully, would benefit the patient and the society. And to give you just some figure, partly because I just finished the annual report recently, but so this figure, it just shows that in the past year, what have we been doing? We have supported 245 study. We recruited 2,202 patients, in total of nearly 5,400 visits. We have 53 staff, not all of them working full time, and we have an annual budget turnover of about 1.5 million. And over the last year, we have been able to leverage additional 4.4 million in research income, about a quarter of those coming from the industry. Now, that's all seems singing and dancing, but of course, it's nothing to do with me because I've only been in post for the last few months. So it's all down to the hard work of the facility and my predecessor, Professor Walker. But we do still face a lot of challenges. And I think these are one of the things that we need to think about is, these are just some of the example, not all. So we need to think about how we can involve and engage patients more better. And of course, there's a lot of new development and new treatment being developed. So how do we respond them to the rapidly evolving therapeutic landscape? And some of these new treatment are getting more and more complex, for example, gene therapy and cell-based therapy. So how do we support those? And of course, we don't have unlimited resources. So we need to think about how do we prioritize the resources and also, secure additional resources as we can. And of course, it is important to maintain an up-to-date knowledge to make sure that we can run this study in a high quality and effective manner. But at the same time, we also need to work effectively with the industry and clinical research organization so that we maintain competitiveness, because we are not the only CRF in the country, of course. Now, but I wanting to focus a little bit about the patient involvement and engagement challenges here because of who we are speaking to today. So one of the things is about how do we promote the research opportunity to more people and participants. And partly because at the moment, because these are very specialized study, very complex study. So currently mostly approached by the investigator. And so that may restricting to the patient who are not attending our center may not necessarily know about this. So how can we make sure that people know about this? Could we use a registration like the Research Plus Me that the trust has set up for a later phase study? And then I think, how do we communicate effectively and honestly about the concept of experimental medicine and early phase clinical trial? Because there's certainly a lot more unknown and known risk. And, and we know that at the end of this trial, we can't continue to provide a treatment because of the regulation reason. How do we explain the necessary to use placebo, i.e. a dummy drug? And of course, because if we are the center that who can do the study, then we need to think about the potential patient burden. So for example, people who live a long distance from us may be potentially disadvantaged because it's harder for them to travel to Newcastle to do this study. So whether we can consider a hub and spoke model where some of the risk visit can be done more locally. And of course, because of the time burden, I mentioned that a lot of these studies take a lot of time and number of visits. So we may make people who have working or other responsibility harder to join. So whether we need to think about whether we can run some of this study out of hour, but it's not that simple because there's logistics, support and safety, all kinds of issue. And then also, how do we involve and engage people from underserved area, different ethnic background, different educational level, because they may need different way to explain the concept of this trial to them. And then I think we also needed to think about how do we engage the public, because these are early phase study, a lot of them we don't really know for sure whether they work. So typically the figure quote is actually only about 15 to 20% of the early phase study actually shows promising enough results to take it forward to later phase study. So, people may ask, so why do we spend so much money that, 80 to 85% of the time, the money you could say potentially wasted. Now, but finally, I just wanting to talk about the vision. So our vision is that hopefully we can make experimental medicine and early phase clinical trial more accessible to everyone and being inclusive. We're wanting to continue to expand our research portfolio because there's so many new treatment, being developed in so many different disease area. And this facility has been set up 15 years ago. I think it's time that we need to think about how we can modernize and upgrade some of our facility. We must think of a good pathway to continue to support training and education in clinical research. And that's not just about our own staff, but also about the clinical staff, other people, to encourage their interest in doing clinical research. And as you see that we have a large number of partners, how can we strengthen the collaboration with all our partners as well? And ultimately, we hope that we will become the go-to site for experimental medicine study and early phase studies. And in other words, we will be one of the best CRF to accelerate translation of scientific discovery to the clinics, and hopefully will address the pressing health needs and also benefit the patient and society. So I just want to end on saying thank you, and there's no doubt thank you very much for all of your attendance today and listening to my introduction. But of course, we must also thank the NIHR, which provided us a substantial proportion of our operational costs for the last 15 years, and also some of the organizations who had helped us initially to set up the facility. I'm afraid 15 years ago, I wasn't here, so I might miss out some organization. If any of you representing those organizations, I apologize if your name are not specifically mentioned. And I would encourage you to go and check on our website, which Dominic is currently updating. So don't look at it yet, maybe come back in a couple of months' time. And if you like, you can follow us on our Twitter. So thank you for your attention. So I'm going to stop sharing, although I need to find how do I do that. So give me a minute, or maybe Charlie can just stop me from sharing.
That's all right, I've stopped you there. Thank you so much for that. I think it was a brilliant introduction to get us going in today, to introduce us to the clinical research facility, and a brilliant way to start off today. I can see some questions coming through in the Q&A, and Dominic's been doing a brilliant job of answering some of those. If you have asked a question, they've been answered. So if you just go back into Q&A and click answered, you'll find an answer to your question. We are going to move very swiftly on now to our next speaker on the agenda. So I'm going to hand over to Dr. Christopher Duncan, who's going to talk to us a little bit about how Newcastle has been supporting some of the COVID vaccine trials over the last year.
Great. Can you see this okay?
Yes, we can.
Excellent. Right, fab, thanks. It's a great privilege to be invited to talk to you today and to share a bit of our experience of doing COVID-19 vaccine trials in the CRF. I'm Chris Duncan. I'm a clinician in infectious diseases in Newcastle hospitals, and I'm also an academic in the university. So as you probably know, we were involved in the management of COVID patients and have been since really the beginning of the pandemic. And it became increasingly apparent to us that research in general and research into vaccines in particular would be crucial in finding a route out of the pandemic. The purpose of vaccines effectively is to mimic the immune response that's mounted to natural infection without somebody having to actually experience infection. And the nature of the immune response that is generated by vaccines constitutes both things called antibodies, which act to block entry of viruses into cells and immune mediators called T cells, which not only help antibody production, they also kill infected cells and help to resolve an infection. And the target of COVID-19 vaccines is a protein in general on the surface of the virus itself, the SARS-CoV-2 virus, called the spike protein, and that's the little blue knobbly bits on this cartoon of the virus here. You can think of this protein a bit like the key that the virus uses to get into cells. So blocking its function is obviously quite important in the mechanism of vaccines. And the platforms that are being used and are under development are kind of fairly diverse, from the more traditional vaccines that we are quite accustomed to, involving use of purified proteins or inactivated whole viruses, to the much more novel and very exciting technology that's really come to the fore in the last 6 months during development of COVID-19 vaccines, and that includes mRNA technology and also the use of viral vectors. And basically these methods are designed to introduce a tiny little bit of genetic information into cells that leads the cell to make spike protein, and that's the focus of the immune response. And as Fai has already kind of illustrated, in drug development in general and in vaccines specifically, there are a series of steps that we go through in doing clinical assessments from the stage of having a candidate vaccine to the point at which that vaccine is deemed to be ready to be rolled out and used against the disease. And these are just summarized on this slide. From the very early initial first-in-human phase 1 trials that are done in very small numbers of people and focus almost entirely on safety, to the much larger phase 3 trials, which, as well as studying safety and the immune response induced by the vaccine, also focus on whether or not the vaccine is doing what it's supposed to do. In other words, is it effective? Does it have efficacy in protecting against the disease itself? And to address that question, we need much larger numbers of people involved in those trials. So in the tens of thousands. And so what I'm going to talk to you today about is a couple of the COVID-19 vaccine studies that we've been doing in Newcastle, which I've been principal investigator on at both ends of the spectrum. I'm actually going to focus on the Oxford vaccine study that we started doing almost a year ago to the day. And I'll talk a little bit about a phase 1 trial that we started in the new year. So everybody's probably heard of this vaccine trial. It's been very, very well covered in the media. So this is a phase 3 trial involving 10,000 participants across the UK, about a fifth of those aged over 55. And there were 20 different sites involved in this phase 3 trial. In addition, there were sites in South Africa, in Brazil, and also in the US. And it was testing what's now known as the Oxford AstraZeneca vaccine, which has a bit of a mouthful, ChAdOx nCoV-19 or ChAdOx-S. So this is the Oxford AstraZeneca vaccine. And participants in the trial were randomly allocated to receive either the Oxford AstraZeneca vaccine or a comparator vaccine against meningitis ACWY. And the participants in the study didn't know what they'd been given during the trial, nor did any of the clinical team that were looking after the participants and making assessments of them if they developed coronavirus symptoms. And that's known as blinding, and that's an important part of conducting studies so that bias isn't introduced either in the reporting of adverse events or the assessment of them. So the participants in the study had multiple different visits over the course of their participation. In general, they had 2 doses of either of the vaccines. And if they developed any symptoms of coronavirus at any stage, they got in touch with us and were reviewed. So what did we actually do? Well, we recruited nearly 700 participants, which was the 3rd top recruiting site in the UK. So effectively, 1 of every 14 people in this trial came from the Northeast. These participants came to the CRF and had over 4,000 visits, and we assessed over 140 of the participants in the trial who presented with symptoms of coronavirus. And the participants themselves administered over 30,000 self-taken PCR nose swab tests on a weekly basis during their participation in the trial. We had over 70 staff working directly on the trial over the course of it. And importantly, given the scale of that effort, we maintain the quality of the research based on assessments by the regulator and also by the Trust's research assessment teams. And importantly, the participants who were involved in the study had a good experience of the trial, and that was reflected in excellent participant feedback through the NIHR system. And really the only way that this was possible was just through an enormous amount of teamwork across a number of different partner organisations that were crucial to delivering this study, and they're summarised here. And effectively, every single piece of this jigsaw was critical to getting us to where we got to. And there's lots of additional groups that are actually not represented on this slide, just because it was such an enormous team effort. So this slide just summarises what the trial actually showed, and I'm not going to go into this in great detail because it's all been published and well publicised, but I thought it'd be useful to review anyway. So the side effect profile of the vaccine was as we expected it to be from the earlier phase 1 and phase 2 components of the study. So between a third and a half of the participants experienced flu-like symptoms after vaccination, which settled quickly and were improved with simple measures like paracetamol. There were no serious safety concerns during the trial. The vaccine induced a robust immune response, both in terms of T cells, not shown here, and in terms of the level of antibody, which is shown here. And after 2 doses of vaccine, participants mounted an antibody response that was equivalent to that measured in patients who have recovered from coronavirus infection. And that translated into a very good level of efficacy against COVID, with 70% protection against symptomatic infection. And importantly, no participant in the trial developed severe symptoms or needed to be hospitalised with COVID-19 who received the Oxford AstraZeneca vaccine. So the efficacy against the more severe end of the disease spectrum and the part of COVID that caused us such problems in the NHS over the several waves of the pandemic was extremely good. So as a consequence of that, the vaccine was approved for emergency use by the UK regulator, the Medicines and Healthcare Regulatory Agency, just towards the end of December, and then started being rolled out through the NHS vaccine rollout in the beginning of January. So what sort of impact has this had? So we're now kind of nearly 6 months from that approval. So certainly in the UK, as we all know, the vaccine rollout has gone extremely well, and over 60 million doses of coronavirus vaccine have been given. Approximately half of those have been the Oxford AstraZeneca vaccine, the rest would be the Pfizer-BioNTech and Moderna vaccines. The Pfizer-BioNTech mRNA vaccine was approved about a month or so before the Oxford-AstraZeneca vaccine was. So that was rolled out first. And as a consequence, over 70% of UK adults now have antibodies to the coronavirus. The vast majority of these induced by immunisation, only about 15% induced by previous infection. And Public Health England estimates are that the NHS COVID vaccine programme has prevented approximately 10,000 deaths and over 35,000 hospitalisations, which is obviously a massive impact on the pandemic that we've seen locally. But the strength of the Oxford AstraZeneca vaccine, I think, is reflected in its global reach, because this is the vaccine that has got the largest global reach of all of the coronavirus vaccines that have been developed. So over 400 million doses of this vaccine have been delivered worldwide to 160 countries. And importantly, 2/3 of those countries are lower middle income. And as we know at the moment, in serious need of access to vaccines. And you may have heard of the COVAX initiative, but this is a global initiative to try to get vaccine to countries that are most in need due to their socioeconomic status. And of the 67 million doses delivered through the COVAX initiative so far, the vast majority of those have been the Oxford AstraZeneca vaccine. So it's really having a major impact globally. As part of the NHS vaccine rollout, Public Health England is monitoring the effectiveness in the real world of the different COVID-19 vaccines. And so these data are taken from the most recent vaccine surveillance report by Public Health England. This is quite a complicated set of data, but effectively what it appears to be, what we appear to be seeing from these real-world effectiveness data is that either of these vaccines are performing to a high level, and it seems effectively that they're interchangeable. So, you know, it doesn't really matter which of the two vaccines one gets. They're both performing very well against various outcomes associated with COVID-19 infection. So I'll turn very briefly to mention the phase 1 trial that we've been doing, because that's much more aligned to what the CRF is core business is all about. This vaccine is being trialled in the UK through a partnership between the UK government and the manufacturer. And so there are 4 UK sites that participated in the phase 1/2 component of the study, including ourselves. As is always the case with the COVID-19 pandemic, the CRF team set up this trial over the Christmas holidays, and we were set to recruiting participants during the 3rd wave of the COVID pandemic in January. Despite that, we were the first site to recruit to target, and this vaccine looks promising. It's got very good tolerability and generates a good immune response, and has progressed now to phase 3 trials, which are being conducted both in Newcastle and in Northumbria in the northeast. So just at the end, want to briefly reflect. As I said, we're about a year now since we screened the first participant in the Oxford study, and I wanted to kind of think about what lessons I've taken from this experience. I think one of the key things is the importance of shared purpose between the participants, between the study teams, and partnership between the university and the NHS. I think results in incredible progress being made. But it's important, I think, also not to forget that this progress didn't come out of the blue, and we should recognise the decades of underpinning basic science that has led us to this point. The mRNA vaccines and the viral vector vaccines didn't just magically appear at the beginning of the COVID pandemic. They were based on lots and lots of work that went on prior to that. So I'll close there. This slide just summarises the many people involved in all this work, and actually doesn't include probably half of the people involved. So I apologise to anybody on the call who isn't referred to here, but I want to pay particular tribute to the participants who stepped forward during a very uncertain period and gave up their time and a lot of commitment to being part of these research studies, and without their involvement this just wouldn't have happened. So I'll leave it there. Thank you.
Thank you, Chris. We've got we've got plenty of questions that have come in, so I'm hoping to run through a few of them. I don't think we'll get all answered, so if you can hang on the call for 5 minutes and just sort of type a few answers out, that would be great as well, Chris. So the first one is, how did you find being involved in these trials when obviously so much hope was pinned on the Oxford AstraZeneca vaccine coming to fruition, shall we say? Did that add more pressure to your role? Sort of, how did you feel about it being involved?
Yeah, I mean, I kind of probably can only talk for myself. I don't want to speak for the massive team that was involved, but I mean, I think it was the main feeling of being involved was just a privilege. I think it was really important, and everybody in the team was sort of unified with that sense of common purpose, and it was great because I didn't mention it, but the team that we sort of put together who helped deliver this came from all over the research and clinical parts of the organisation, and everybody just were kind of united in doing this. So it was a great experience. Actually, it was an honour to be involved.
I think we all thank you and the team for everything you have done as well. So another question there, what are the advantages of having additional sites in South Africa and Brazil?
So I think that was a decision made by the sponsors, and at certain points in the UK, the prevalence of coronavirus fell dramatically. So after the first lockdown, there was a period of time when there was very little infection before the second wave happened in the north of the country, and then our third wave, but which gets referred to as the second wave. So there were periods of time where there wasn't very much coronavirus circulation, but there was a lot more in other countries. And so one of the reasons to have trials in multiple countries was to try to get to an efficacy signal as quickly as possible, because that was important to see whether or not the vaccine was going to be effective, and to try and get that answer quickly. The other reason I think, which is important, is to understand its performance in different populations, particularly when the aim was always at the beginning of the whole process to make a vaccine that was going to be of benefit to everybody and not just to people in the UK.
And then I think a final one before I'm going to have to hand back over to Charlie. What would have been the roles of the medical doctors in the Oxford trial? So
primarily consenting participants into the study, so going through the information, discussing the risks, discussing the processes, so that people were fully informed and consented, and then doing medical assessments to ensure that participants didn't have any health problems that would have put them at increased risk of participating. And then the medical teams were involved also in assessing patients who, participants who developed symptoms of coronavirus, because everybody who did so came up and had a full medical assessment, so that we could determine A, whether they had coronavirus or not, and B, whether they needed any additional care.
Thank you again, Chris, and I'll hand back over to Charlie to introduce the next speaker.
Thank you so much. Like Dom says, there are a couple of other questions in the Q&A, so I'm sure Chris and Dom will be able to get around some of those and get you the answers to those questions. But we're going to take a little bit of a detour now into a different area of clinical research, by handing over to Richard Holliday from the School of Dental Sciences, who's going to talk to us a little bit about how they've been making dentistry safer during the pandemic.
So good afternoon, everyone. Hopefully that's my slides nice and clear.
Yes, that's
good. Perfect. So my name is Richard. I'm essentially a research dentist based in Newcastle. So we work in the dental hospital. It's also the School of Dental Sciences, where we train dental students and dental hygienists and therapists. So I'm going to take you through a bit of a whistle-stop tour of what's happened in dentistry during the pandemic, how we're now operating, and how our research in Newcastle has helped us get back to kind of what we're doing now more safely. So you already heard quite a lot from Chris about the SARS-CoV-2 virus that causes COVID-19. I'm not going to tell you any more about that, only to say that it's present in saliva when people are infected. And that's a problem for us as a dental team, and you'll see a bit more about that later. So this is probably quite a common sort of image you'll be used to if you ever watch the news. So we see lots of these sort of graphs these days of different forms. I'm just really putting this up as a template to overlay what happened in dentistry at different time points during the pandemic. So the first thing that happened in March last year was that dentists were told they had to close their doors. So every dental practice in the UK got told they couldn't see patients face to face. That happened almost overnight. So that means in our region, about 350 NHS dentists had to close their doors. So quite a considerable issue. And actually they stayed closed for over 2 months and opened again on the 6th of June with some guidance on how they should open in coming literally hours before they opened. Public Health England came in with some kind of further modified guidance in October. And now dentists have been open since. Dentists are working differently. They're currently maybe working at 50 or 60% of what they used to be doing. And they're trying to increase that. So what's the problem with going to the dentist and COVID-19? Well, the first problem is dental teams, so the dentist, the hygienist, the nurse, get close to patients, so they get within that 2-meter physical distance. So that's the first issue. The second issue is sometimes we make quite a mess. So we use instruments that have a water spray. So we might use a high-speed drill. We might use a scaler, so the instruments that clean your teeth. And both these have a water spray. So this spray creates a fine mist, and this mist is sometimes called an aerosol. You'll probably be familiar with this term, and this could pick up virus from the mouth, from the saliva, and spread it to the dental team, or it could stay in the air. So that's potentially a problem. And the other issue is how long does it take to clear that room or that surgery or that space till it's safe for the next patient to use? So these are the 3 kind of fundamental issues we had at the start of the pandemic. The first real headache was all dentists are closed. How do people get emergency dental care? People don't stop getting toothache. They don't stop getting dental infections. And some of these can be life-threatening if they're not treated. So we're quite fortunate here in the Northeast in that we have the dental hospital. We weren't required to close as part of these restrictions. We're very fortunate to be part of the Newcastle Hospitals Trust, and we had all the appropriate supplies for PPE. So overnight, we essentially turned into the only dental provider in the north of England, providing care for over 3 million people. So we had to set up a service very rapidly. There was guidance that was changing, day on day. So it was quite a busy time. We had the normal sort of restrictions you would have imagined, but was able to provide care for several thousand people during that time. We also collected data on what we did during that period that was fairly interesting. So here's kind of 2-week blocks. So the first 1 or 2 week on the left here. Let me just get my pointer up actually. Yeah, so the first 2 weeks here, the middle 2 weeks, and then going up to 6 weeks, week 5 and 6. So the H here marks the dental hospital in Newcastle. And this is a heat map showing where our emergency patients attended from. So the green and the yellow show some of the more dense areas, and the red shows where some patients have come from. You can actually see by the middle 2 weeks, we were serving quite a large area. So patients were traveling sometimes 200 miles to come to the dentist, right from the West Coast, from Scotland, from down the Leeds and York. So quite considerable distances, but luckily by about week 5 or so, the back end of week 5, some of the other urgent dental care centres could come on board and start providing face-to-face care. So all these crosses represent some of the other dental practices that have been able to come on board and take some of the pressure off. I must say, kind of regular dentists, high street dentists, were working during this period. They were busy providing telephone advice and providing prescriptions via pharmacists, but they weren't allowed to see patients face-to-face. So we wrote up our experience. It's actually the first detailed report of kind of providing this sort of service and our learning from it that was reported actually anywhere in the world. And over the last year, it's been the most cited, the most referenced journal in the British Dental Journal. So it kind of shows how hopefully important our work was and how much it's been used by other people. So that was one aspect of what we were involved with, kind of providing care in a safe way for the population. But the other aspect was the researchers in the team started to kind of scratch their heads and think, well, what research is there on this topic? And actually, a lot of it's quite historical. A lot of the classic papers in this field, a lot of the classic studies are from the 80s, some from the 90s. Some of them are very good studies for their time, but they're quite outdated now. And you can see a couple of examples of images from these papers that kind of show how dated they might be. So we kind of started realizing there's actually some quite serious unanswered questions in this area, and we started putting in place some of the research to answer them questions. There's a whole range of ways you can start to look at this, but we started using a technique using a fluorescent dye. So this is fluorescent. It's a really pretty dye. It makes really nice colors. It's very safe. So the Environment Agency use it in studying pollution and river studies. But we were using it to model a virus, and you can use various techniques to detect this, and we can kind of see where this might go inside an environment and how we can reduce its spread. So this is me during the first lockdown, and this is one of our training suites in Newcastle where our dental students learn to do their dental procedures. You can see I'm doing a dental procedure on this pretend patient here, and there's this spider's web around me of samples, and we're kind of trying to understand here where the contamination goes from the patient, how far it goes and in which direction, to try and get a really detailed understanding of this. We developed two models for this really. So the first model was where we put the dye, the fluorescent, into the drill or the scaler. So you can see in this video here, the water spray is coming out and it's got fluorescent in it. So you can kind of model where it goes now. So you can see my chest here, my left chest and my left arm is getting quite contaminated. In this image, we're using some suction, and that seems to be relatively good at reducing that in some way. This is not representative, though, of the real situation. This is just showing us where the water spray goes. It's not really showing us where potential virus is going. So we developed a second model. This second model is more kind of relevant to real life. This time, dye is introduced into the mouth to represent saliva. So there's a slow pump that's slowly injecting virus through these little tubes, and you can see some's just come in around these front teeth, and this person's doing a procedure on a front tooth with clean water coming out the drill. And this study was looking at how much of this dye is picked up in the water spray and transported maybe to this filter paper here. So we developed a whole range of methods for measuring this fluorescence. So one was to look at filter papers and then come up with a fairly sophisticated way of washing off that dye and then measuring it. So we had a whole lab team behind us processing these samples. We also developed ways of measuring the aerosol that was left inside the air and that left floating around. So this is an air pump that was measuring that. We also have a series of these optical particle counters. So these are usually used for measuring things like environmental pollution, road traffic pollution. But we can use them in some settings to try and understand more about the aerosol. They're very, very sensitive. So essentially we have different data sets that are being triangulated to give us kind of a single answer. So we've investigated a whole range of things that I couldn't possibly go through this afternoon, but I'm just going to pick out a few of the key highlights. So one of the main findings was we now started to get a much better picture of relative risk. So to understand which dental procedures were more risky and which were less risky. So here's just two examples. So on the left here, we have someone having their brace taken off and then a polish of their teeth. This is a heat map. You'll see a few of these in the next few slides. And these little dots represent where we've taken samples from. So this is the spider's web you saw earlier. This is our patient in the middle. This is the dentist or the hygienist or the therapist. And this is the dental nurse. So you can see for this particular procedure, so taking a brace off, there's actually not much contamination. There's a light blue color here, and then some very faint colors here and here. It's very different to the situation on the right. So this is where we're preparing a tooth for a crown. We're using a high-speed drill for this with water spray. And you can see there's some very dark blue colors here, which indicates a quite heavy contamination. And they go fairly far out as well. So this shows we've done this for a whole range of procedures, and we can kind of see what's more risky, what procedures we have to be more worried about, and which procedures we don't potentially have to worry about so much. We also wanted to know how we can reduce the risk. So there's 3 ways of doing this, really, that we've looked at. The first is to reduce the production in the first place, so you don't make as much aerosol and contamination. The next issue is, if you do produce contamination, can you remove it close to the source to get rid of it, nip it in the bud almost before it's spread into the room? If it does spread into the room, how long do you have to leave it to settle for before it's kind of safe for the next patient to come in? So they're the 3 questions we've been looking at. So one idea to reduce kind of the production is to redesign the drill. Do we really need this really intense water and air spray that comes out of the drill? This is the classic drill, the high-speed drill. Same sort of heat map as before. You can see it produces a lot of contamination around this area. This is the slightly redesigned drill. You can see it's less, but it's still got some green colors further out. And this is another modification to the drill design. And you can see now there's hardly anything beyond the immediate area. So this is a much safer configuration of the drill to use from a kind of a cross-infection point of view, from some very simple modifications to design. The next thing we can do is, if there is aerosol produced, how can we remove it or reduce it? So one thing we can use is use the standard dental suction. This is what we've used for many, many years. You'll be very used to this. It's like the little hoover that the dental nurse will use inside the mouth. And this is actually very effective. It reduces and removes most of the aerosol. So that is something that's very simple. It's something we use all the time. The other thing we could do is essentially have a device outside the mouth, essentially a big hoover, and some of these devices have been developed. And you can see here the aerosol's been picked up and taken into this device. Something very specific to us in a dental hospital environment where we train students, and this is the same in all the the dental hospitals across the UK and the world, is that we have these open plan clinics. So students can be supervised in an open space and learn. So we had some very specific questions about this environment, and we conducted a whole range of studies in this setting. We essentially found that dental suction, again, is really powerful. It's a very powerful tool and reduces contamination considerably. And we also looked at kind of safe distancing, and we found that at a 5 meter distance, we essentially couldn't detect any of the contaminated aerosol. And if we could detect it, it was diluted very highly. So around 60 or 70,000 times diluted. So it gave us a lot of confidence that a 5 meter spacing was a safe setting in this environment. So we have some ongoing work actually using a virus now. So we're using something called a bacteriophage. This is a safe virus. It's actually a virus that doesn't infect humans. It only kind of affects bacteria. We're using one that looks very much like the COVID-19 virus. And we're replicating some of our experiments now using this virus. And this is our particular virus here. Here's a petri dish that has got essentially virus food on it. And every drop here is a viral virus that's kind of landed and has formed a little colony. And this is from one of our mock experiments. So there'll be some work on this out shortly. So where is dentistry currently in the UK? So a whole range of guidance documents and procedures for how dentists should operate were produced, and a lot of our work inputted into these. Dentists are getting back up to speed, but it is tricky. They still have quite strict criteria to work to, maybe working at 50 or 60% of their normal workload at the minute. And you might have seen some of the news stories this week about how difficult it has been for some dental practices, and particularly from a business point of view, whether they're going to stay operating or not. So it's a tricky time for dentistry. let's just think about if you were to go to the dentist today or tomorrow, how might it be different? So the first thing is there'll be some very simple things, like the waiting room would be spaced out. There'll be a few people in the waiting room. You would have been potentially screened. You would have been screened before you come to the visit. You would have maybe had your temperature checked. When you visit the dentist, they may wear fairly normal personal protective equipment if they're just doing an examination. But if they're doing a procedure that makes an aerosol, so uses a water spray, they may have to put a gown on, they may have to have a special mask, they may have to have a visor on. And there'll be a whole load of things you won't see behind the background. So they'll have thought about the ventilation in the room, and they'll have thought about when it's safe for you to go in, when it's safe for the next patient to go in. So there's a whole range of things going on in the background. So what do we know a year on with regards to dentistry? So we know that actually it's fairly minimal risk with the current mitigations. There's been no known cases of COVID transmission in a dental setting. The simple stuff is very effective. So the things we do already, so dental suction, rubber dam, are very, very useful. There's a large diluting effect. So the water spray, the aerosol that we produce in dentistry is actually mainly water, and it contains a very small amount of potential saliva and then potential virus. We've learned that ventilation is key, just as we've learned more broadly. It's been introduced into the hand space saying with regards to fresh air. It's really the same in dentistry. And with regards to the future, I think there's a lot of potential technological advances that can happen to reduce the risk with regards to dentistry. So as the others have mentioned, there's big teams behind all of these sorts of projects. So I'd like to thank James and Chris, who've been joining and leading this for me, and to the huge range of co-investigators. I see a couple of them on the call today. So Kimberly, one of our research nurses, is on the call. And huge thanks to the collaboration really between the hospital in Newcastle and the university and various organisations who have funded the work. So thanks for listening. If you want to read any more, we have our papers on online blogs. So you can take a picture of this QR code and it will take you to that. And I'm happy to answer any questions. Thank you.
Thank you, Richard. Yeah, there's been a fair few questions come in. I have tried to answer ones where I can, but I've got about 6 for you so far. So this is more of a comment than a question, but it's interesting obviously to see the range of clinical trials you've all been involved in. This individual struggled to get a dentist appointment in the first lockdown and would have understood a bit better if they had heard your talk first. So
that's interesting, yeah.
The second one is, was it an eye-opener, all of the aerosols being produced to yourself? Did you find it? Obviously, you would have known that it was a potential, but was it eye-opening, how far it went, etc.?
It was a bit, we haven't really considered it a huge amount before. It's not been a problem in dentistry. Obviously, we have very strict cross-infection in dentistry, but it's largely been in regards to bloodborne viruses, so HIV, hepatitis. They were the big issues in the 80s and the 90s. And a respiratory kind of pathogen hasn't been a huge issue before. So that kind of is why the research is so old on this topic. Yeah, and the fluorescent dye is really neat to work with because it produces great visuals. So we're able to kind of shine the light that illuminates it on us afterwards and see the contamination. So it's a very useful training aid and a prompt to the dental teams about how important it is to have a really good technique on how they take off the protective equipment, for example, afterwards and not contaminate themselves. So yeah, it was eye-opening, yeah. And
then another one, what happens to the aerosol when it has been picked up by the new device?
Oh yeah, so good point. So the device, the huge Hoover thing, I think that's probably the thing they're talking about. This has a HEPA filter inside it and a UV light as well. So the virus is essentially deactivated and then held within the HEPA filters. Sometimes it's actually pumped outside into a safe space as well once it's been deactivated. So yeah.
And then probably one taking you back a little bit, what can an aspiring dentist take home from the presentation that you've just delivered?
Get involved in research. So we've had a huge team involved. We've actually had some students involved who've loved it. We've had dental students, therapist students. Yeah, so it just, I guess the variety of what you can do. So, there's been kind of engineering aspects to this. We had to build a rig. We had to go to a DIY store and build the stuff. We had to learn about microbiology. We obviously have some experts on the team, but I've had to kind of learn about all these different aspects. So it's about kind of problem solving and yeah, getting involved in research. So that's what I recommend you do.
So I suppose that leads me quite nicely onto this question. Could this research lead to other work around infectious diseases, or is COVID the main focus?
No, it's a much bigger focus actually, and James Allison, one of my one of the leaders of this project with me, is actually taking it on as his PhD project, looking much more broadly than COVID-19. So looking at how we can better design dental surgeries in the future, how we can, update some of our instrument designs so they are potentially less of an issue for future pandemics, whatever they may be. So getting us ready for the future.
And that's all the questions that we've got in there at the minute. Okay. There is one about continuing safety of medication, but I'm hoping Fi can chip in with that one. Because I think it's one of the biggest talks. So there we go.
Thank you.
Thank you very much, Richard.
No worries.
Thank you so much for that, Richard. I think that was a really, really interesting talk, and I personally really enjoyed it. So I'm hoping a lot of the people on the call enjoyed it as well. We're going to take a little bit of a break now because we're conscious we've had you in front of a screen for a long time, and we want to give you the chance to get away, to get up, to get moving. So we are going to take a quick 10-minute break. Don't leave the call, don't go anywhere, but we do want you to get up, get moving, do a lap around your house, get a cup of tea, go to the loo, do whatever you need to do. But don't sit in front of the laptop. This is you've got permission now to leave the screen, get away from the screen, and go and do something else for 10 minutes. And join us back here at 3:15, where we'll be starting with our next talk, which we'll introduce when we get back. So don't go anywhere, but also do go somewhere away from the screen. Okay, we'll see you soon. Okay, hello everyone. I think we're back now. I hope you made good use of your 10-minute break there. I did run around my house, went to the loo and got a new glass of water, which I think is a relatively productive 10-minute break. We're ready to get going again. I'm hoping that the majority of you are back in the room. That is the one thing with Zoom webinar, because I can't see you. I don't know how many faces are back, but I'm hoping that you're all back in the room with us now. So we're going to move swiftly on now to our next presentation from Professor Nicola Pavesi, looking at understanding the needs of Parkinson's patients during COVID-19. So a slightly different aspect of clinical research in Newcastle. So I'm going to hand over now. Thank you very much.
Thank you, Charlie. Good afternoon, everyone. And let me start my presentation here. So my name is Nicola Pavesi. I'm professor of clinical neuroscience, and I'm a consultant neurologist with a special interest in Parkinson's disease and other movement disorders. That is both a clinical and research interest. So So I'm sure you're all familiar with Parkinson's. Is a progressive neurodegenerative disease. Is the second most common neurodegenerative disease in the world after Alzheimer's disease. And in UK, about 150,000 people suffer from Parkinson's. Unfortunately, we still don't know the cause of this condition, but what we know now very well is what's happening in the brain of a patient with this condition. There is the accumulation of an abnormal protein, which is called alpha-synuclein. And you see that is aggregates within the cells of the brain, the neurons, because making this formation that are called Lewy body disease. And this leads to a loss of cells in the brain, particularly in this specific area of the brain that is called brainstem. And you can see that normally this area is very black because there are a lot of cells that have a pigment in here, while in patients with Parkinson's disease, this area is not as dark as in the normal subject, that's because there has been a loss of cells. And these cells are very important because produce a chemical called dopamine that is very important for the movement. Parkinson's is a number of symptoms that are involving the movement, so they are called motor symptoms, and these are the tremor, the stiffness in the muscles that we call rigidity, slowness when people are performing movements, and problem with balance. There are also problems that are not really related to movement, but are also very common in people who have Parkinson's, and that can be depression, sleep problems, dementia. But those are only some of the problems, because Parkinson is a very complex disease. It's also a disease that runs for 10, 20, sometimes even more years. And you can see that what we now know is that when patients are diagnosed, so when they start having the classical motor symptoms, they might have had the disease for several decades with other symptoms like constipation, loss of sense of smell, problems with sleep, depression, and so on. And you can see that as the disease progresses, the number of symptoms, motor and non-motor symptoms, become more severe and more frequent in these patients. Unfortunately, at the moment, we do not have any medication to slow down the progression of the disease. But there is a lot of research ongoing for trying to better understand the Parkinson and improve treatments. And Newcastle is really very much at the front line of Parkinson's research. You can see this is a Medline research, so it's a software that we have to look at papers that have been published on the topic. And if here I'll put two keys, Newcastle University and Parkinson's disease, and you can see that in the last 10 years, 337 papers have been published by researchers in Newcastle on Parkinson's disease. So this is very, very impressive. In CARU, the clinical aging research unit, we have a lot of number of clinical trials and studies for patients with Parkinson's disease at any stage of the disease. And we also have clinical trial with new medications that are currently under development. And this is great because patients have the possibility to try this new medication very early in the stage of their development. And this medication mostly aim to slow down the progression of the disease. We also have a very special brain scan, the PET scan and the MRI scan that let us to see the structure of the brain, but also the way the brain works. So for example, these are examples of scans. You can see that there are different types of scans. We can look at the different chemicals in the brain, the dopamine, the serotonin, the acetylcholine. You might be less familiar with that, but it's important for cognition. And you can see that all these scans enable us to understand the differences in patients with Parkinson. For example, you can see in these scans that people with Parkinson have a reduction of the dopamine cells in this specific part of the brain compared to the normal. There is a reduction of serotonin in this part, which is the brainstem, and also a reduction of acetylcholine in these parts of the brain, this is the cortex, which is involved with cognition. So you can see that we can really try and understand what is causing the symptoms in Parkinson and possibly find targets for new treatment. More recently, we have joined the Parkinson Progression Marker Initiative. This is a large study, is a very landmark study in Parkinson. The idea is to discover, identify biomarkers of disease progression that can help the development of new medication. And we are doing very well with recruitment of patients, but also healthy volunteers. So there is always in our studies a room also for healthy volunteers that want to help with research. So as you can see, a lot of research going on. And unfortunately, the COVID has had an impact on the patient with Parkinson. This is again the research in published literature. I'll put here Parkinson and COVID-19. And you can see that in the last 2 years, there have been 354 papers published on Parkinson and COVID. And you can see that in 21, it's only 6 months, we have already topped the previous year. So that I expect will be even more. So a lot of clinicians, a lot of researchers have looked at the impact of COVID in Parkinson's. This study, I thought it was very interesting. It's a very recent publication by Artusi and colleagues. What they did, they review all the literature, looking at the most significant papers that have been published with the topic of Parkinson and COVID-19. And mostly they used the studies that have the largest number of patients. So they in total have found 16 studies who looked at 11,000 PD patients, and 1,000, just about 1,000 of them had a confirmed diagnosis of COVID-19. So what did they find? So they found that unfortunately 28, almost 30% of these patients required hospitalization due to COVID. There was a worsening of the disease. So almost 40% required an increase of the medications because the symptoms were no longer controlled. Unfortunately, 19% died. And the fatalities was higher in the Parkinson's disease patients that were COVID positive compared to the general population. So unfortunately, patients with Parkinson have been badly affected by the COVID-19 pandemic. And that's, this study would suggest that there was a good rationale about shielding and protecting a patient with Parkinson. But also there are other data. This is an audit that was performed by Victoria Foster in my team and Jacopo Pasquini. So what we did here, we audited 62 patients from our clinics with Parkinson to reduce face-to-face exposition. That was a telephone survey. And we asked about physical activity, new or worsening symptoms of PD and presence of motor complications. And what we found that as a result of the lockdown, almost 60% of patients had a reduction in a decrease in physical activity. More than 80% of patients had a subjective worsening of the symptoms of Parkinson, particularly tremor, problem with balance and gait, but also non-motor symptoms like depression and anxiety. And what we found is that there was a correlation between the worsening of the symptoms and the decreased physical activity, meaning that the greater the reduction of physical activity, the more the worse was the worsening. So we have started encouraging people to do physical activity, even if they don't go out at home, to do exercises, even sitting, but moving, because that is very important. Unfortunately, the lockdown and COVID has also somehow affected the research, and we know that the research is being engaged in research is very important for patients, because from experience we know that people who are involved in research tend to do better. The disease seems to be better tolerated, and people engage much better. So how have we met the need of a Parkinson's patient during the pandemic? First of all, we were very blessed with the location in Cairo. You can see we are separate from the main RVI hospital. We are surrounded by parks in here. Cairo is at the very end of the campus. There is a lot of parking space. So maintaining social distancing has been very easy. And there was very low risk for the patient. So we have continued with the majority of our studies. And this has been very good for the patient. We also, both in clinic and in a research setting, we have modified the way we interact with patients. We have offered telephone consultation, also remote visits. So people with internet, there are softwares like Attend Anywhere, where we can actually see the patients remotely, discuss and perform some visits. However, there are some problems related with that. For example, there are problems with connection. Technology can be a problem for many patients. Not all the symptoms can be detected. For example, to assess rigidity, we really need to feel it. You don't see rigidity in the muscles. A balance as well need to be assessed. So some of the symptoms cannot be checked remotely. And then, to be honest, not everyone likes this remote visit. There are people who prefer to interact, to come and talk to us. And so there are a lot of people who don't like the remote consultation. The remote consultation can also be quite long. So in my clinics and my telephone consultation clinics were not any shorter than the face-to-face assessment. So my clinic day was as long as it was in the past, despite many of those were telephone consultation. There has been also changes in the protocols of studies. This is the ADEPT study. It's a study that we are going to start very soon. It will try to understand what's the best medication for people with Parkinson who have depressive symptoms. So 3 different types of intervention for depression. And you can see in the protocol has been adapted in order to have some remote clinics where all the assessment that can be done remotely have been introduced in the protocol. And also the consent process can be now done by phone or video conference. So the patient is sent the consent format with the information sheet, and then we call the patient, we discuss, we talk about the study. So the same time that we offer in clinic, and then if the patient is happy, he signed the consent, is sent back to us, we sign, the investigator signed the consent, and then we send it back to the patient. So a lot of readjustment in order to reduce the face-to-face contact and keep the patients safe. Also, there is a increase on wearable devices. So there are a number of devices now that can be used to assess the movement and the motor performances of patients. These were already developing before COVID, but now there has been an acceleration in that. And you can really see how this can be useful in the future, because they enable to look at the patient at home in their own environment, so not just in the setting of clinics. And you really have some nice information. So this is an example of this device. Is one of the one already on the market. The person wears that the whole day long, and then that is what the clinician receive as a report. So, for example, here we can see at the dyskinesias, that are involuntary movements that people with Parkinson have when the medication, after 5-10 years of treatment. And this is the slowness. You can see this is the time of the day. And you can see that there are some specific times of the day. This is the average of the week where people have more involuntary movements, which means that people are overtreated at this point. The red is when the medication is taken. And then you can see this is the bradykinesia, so opposite. So the lowest is at these times of the day. And that means that this time the patient is undertreated. And this is the medication. So you see that this gentleman is, or lady is suffering from a wearing off of the dose of the medication. You can clearly see that there is a wearing off here. So a lot of information that can be gathered by these gadgets. Said that, is true that I also need to say that, again, many patients don't like it. Sometimes you require a lot of work. This is a watch. Other devices are a belt to look at the walking, the freezing of gait. Sometimes patients need to click buttons or that related to applications. So again, not everyone likes this kind of devices. But I think there is a future for them. And this was my last slide. So just to let you know that there is a lot of research going on at Cairo on Parkinson despite COVID-19. As a team, we are very, very passionate about research, and we are very committed to find a cure for Parkinson, or at least improve the quality of life of patients. And you can be assured that even in this COVID time, we will do our best to provide always the first-class service, both in research and in a clinical setting, in a safe environment. Thank you.
Thank you, Nicola. There is a few questions that have come in, and obviously please keep them coming if we get a chance, we'll get through them all. So the first one, Nicola, is there potential for gene therapy to be a treatment for Parkinson's disease?
Sorry, say it again?
Is there a potential for gene therapy to be a treatment for Parkinson's disease in the future? Yeah,
Parkinson itself is not a genetic condition. The majority of people, there is very often a background or family history, but the number of people who actually carry a gene for Parkinson is about 10%, 10-15% of people with Parkinson's. The picture is very similar, so the symptoms are very similar to Parkinson. Now, the advantage is that we know what the problem is. While in Parkinson, we don't know why the alpha-synuclein is accumulating. Here we know what is starting it, is this gene. And there are medications currently under development to fix these genes. So possibly there will be some treatment in the future. For gene therapy, those, we also intend something else. So we also intend that we can correct something even in people who don't have gene. For example, now we can put, as I said, Parkinson, many of the symptoms of Parkinson are related to the lack of dopamine. So there are clinical trials where you can put in the brain through viruses or the specific cells, some gene sequences that are responsible for the production of dopamine. So these engineered cells will be able to produce some dopamine which is missing. And that is another hope for Parkinson.
Thank you. Another few questions then. So do you think the amount of toxins due to constipation causes the medication to stop working effectively? And if so, if not, should more research be done into this topic?
Yeah, there is research. I fully agree. But unfortunately, all these medications that are now available, that are, as I said, are symptomatic medications. So what they do, they tend to improve the symptoms. They increase the level of dopamine in the brain, are absorbed at the intestinal level. So since this digestion is slow in Parkinson, the problems with gastrointestinal problems, there is a problem with absorption. And that, again, is considered one of the causes of this motor fluctuations, of these complications. Something is already available because now we have some sort of what we called device-aided treatment. So, for example, levodopa, that is the precursor of dopamine, can be infused directly in the intestine. There is a gel form of it. There are subcutaneous preparation of apomorphine. Very soon at CARU, we will start a trial to look at subcutaneous levodopa. So yes, there is a lot of research already going on absorption.
Thank you. And how does the 18.9% deaths for people with Parkinson's compare to other sectors of the population? Do you have any sort of sense of other areas?
I didn't hear you.
Sorry. So how does the 18.9% death rate for people with Parkinson's compare to other sectors of the population?
I think in the population, I think it depends. It's not the same because it really depends on the different population. And there is also been seen that there is a sort of ethnicity thing, but is for what I know, it goes between the 10 and the 15%. But that is data that should be further investigated because I'm not 100% updated on it.
Is there potential for the use of stem cells in the treatment of Parkinson's?
But there is possibly in the future. At the moment, the stem cells are still not clear because one of the dangers is this stem cells can differentiate in something very different rather than in cells that produce dopamine can differentiate in something else. So there is still some risk related to it. There are in the past, there have been a lot of attempts with fetal cells that are not completely differentiated, and they can differentiate in dopaminergic cells, and those have given sort of conflicting results. There is a study currently going on, supported by the European Union, in Imperial College in London. Is one of the main sites with Cambridge University, but the results have not been released yet. That is an option in the future. Hopefully will be rather than, I mean, the idea is either we find medication to stop the aggregation or slow down the progression of disease, or to find what is the first thing happening. So how can we stop the aggregation of alpha-synuclein? I think that is already happening. We have clinical trials in CARU for that. So we have done already a couple of trials. Unfortunately, we're not very successful. But I think I really believe more in these approaches than something like stem cells. Might be obsolete within a few years.
And then another question there. So is the clinical outcome for Parkinson's disease patients with COVID-19 the same across all ages, younger onset versus older people?
No, that study that I've mentioned, they also found, I didn't report that, but they found that the longer the disease, the higher the risk of having side effects and, you know, the complication with COVID-19 and death as well. So it's really disease depending. But, you know, sometimes if you consider that Parkinson is a disease that usually affect people in their 60s and their 70s, the longer disease also mean generally older people. So you need to see what is the Parkinson and what is really the age as well that contributes.
And you've answered another question that was very similar to that as well. So the final one on here, just more from a sort of what do we do in Carew? Do we offer Zoom type consultations, or is it purely telephone calls at the minute? I know obviously there's in person as well, but are we doing any Zoom ones?
We really think that in Carew we have we can do remote visits. So all the clinical trials are now with remote visits. We have this attend anywhere. But as I said, Carew is very safe. We are very, very careful about, the number of patients who come in and our patients really prefer to come and see us and have that extra time and, extra coaching, really like it. But we are prepared to do remote visits, yes, for all the studies that we have.
Perfect. And that's the final question there, Nicola. So thank you very much. And I'll hand back over to Charlie.
Have a good evening, everyone. Thank you for listening.
Great. Thank you so much. That was really, really fascinating and really brilliant talk. We're now going to move on to our last talk of today, which surprises me that we've got here so quickly, because it really has gotten here very, very fast. So we're going to hand over for our last talk to Dr. Emma Rain, who's going to talk to us a little bit about the Bioresource Centre and how they've been ensuring the safety of research during the pandemic.
Thank you, Charlie. Thank you, everybody, for coming this afternoon. It's been fascinating so far, so hopefully this last talk will be interesting enough to finish it off for you. So my name is Emma, and I'm the manager of the NIHR Bioresource Centre here in Newcastle, and I'm going to talk to you today about how we ensured the safety of what we do during the pandemic. But I'm going to have to start to talk about what the bioresource is first, because it's difficult to talk about how we ensured everyone's safety if you don't know what we did. So, and you'll have to forgive me. I think there are some people I've noticed who are definitely participants of the bioresource in the audience today. So if I'm repeating myself, I apologise for that. So the NIHR Bioresource, we're a national project run out of Cambridge. And at the minute, we are over 150,000 volunteers have signed up to the Bioresource. And they sign up, and the idea is that they are willing participants to be approached about early phase clinical trials or research projects, and they're all waiting to be invited. We accept anyone into the Bioresource. That's the beauty of what we do. Anyone can volunteer, be it healthy volunteers or people with certain conditions. And the overall aim is faster access for researchers to those volunteers, better access for participants to research studies that they might not have had access to and they might not have known about. And we want to shorten that gap between the basic research that happens in the lab and genuine patient benefit. So increased, better diagnoses and brand new treatments that we can get into clinic. So we are, as I said, it's a national project across England, and in fact some of our projects expand into Scotland and Wales. There are lots of different centres up and down the country. There are 13 of us, so there's 5 in London, and then the other centres, we're the furthest north, as you can imagine, where we spread out quite nicely. And there are lots of different branches of the Bioresource. So we have a healthy volunteers branch, rare diseases, we have an immune-mediated inflammatory diseases Bioresource, a non-alcoholic fatty liver disease, inflammatory bowel disease. We have a COVID Bioresource, which, as you can imagine, arrived about a year ago. And we have what we call GLAD, which is or our mental health Bioresource, which is looking at the genetic links to anxiety and depression. And one of the fantastic things about Newcastle Centre is that, in fact, we are the only centre to recruit to all of these Bioresources. Sorry, there we are. So just going to quickly talk about exactly how that works. So we have volunteers come in and they complete a health and lifestyle questionnaire. we take a blood sample. That blood sample is sent in the post down to what we call it's the National Biosample Centre, which is in Milton Keynes. We store some of that blood for future research, but we also send that some of it for DNA analysis. And then we use the combination of the data from the health and lifestyle questionnaire that the participant has completed and the DNA analysis, and we match people to suitable research studies. The idea being that it can take a researcher years to build up a cohort of patients that are suitable for their research. And often research studies can be about 3 to 5 years long. So this way, people can researchers can come to us and say, for example, I need 100 people to take part in my diabetes study, and I need them to be between the ages of 45 and 55, and we need a certain BMI, and we need them not to be taking paracetamol. And this is the kind of thing, so we can filter through our 150,000 patients, and we can then invite everyone who's eligible to this study. At this point, it's completely up to the participant whether or not they want to take part. There's no obligation for them to take part in any study if they don't want to. There'll be another consent form, they'll read all the information and find out about the new study. And if they do want to take part, potentially that researcher has suddenly got their entire cohort at once without having to build that up. And it means that their 3 years project can really be spent on research and not about building that up. So that's the overall idea of the bioresource. So now I'm going to start talking about how it was we responded to COVID. So as many people have mentioned so far today, so Richard talking about dentists, all of a sudden overnight we were closed on in March 2020, which obviously was terrible, but very quickly we realized that we couldn't guarantee that this was only going to last a few weeks. We needed to adapt and make sure that we could keep research going as long as possible. So we luckily had introduced electronic consent to the bioresource in August 2019. So this was in an effort to try and be more environmentally friendly, not use so much paper. So we started recruiting on laptops within the hospital. And we were able to adapt our process so that it was no longer just capable of taking place in the hospital on a computer or a laptop. It could be done remotely by the patient or the participant in their own homes. So all the forms could be filled in beforehand. We now call this remote consent. And then we just defer the blood collection for when we were able to come back in to the hospital again, when we were able to open up. And as Nicola mentioned, and I think Fi mentioned earlier as well, the plan, because we work very closely with both facilities, is that our bioresource clinics will be held at the Clinical Aging Research Unit and the Clinical Research Facility. And these are off-hospital sites. They have parking. Nobody has to use public transport. So it's much, much safer to come in and take part in this study based on doing it this way. Instead of sitting around in the hospital for 45 minutes filling out forms and then having blood done, you can do all the forms at home and then just come in and have the blood done at the hospital. So the other thing that I just wanted to talk about is, so that's how we managed to adapt to keeping our everyday research going to our ordinary clinics, as we call them. So we're now fully open and we are continuing with the remote consent because we've seen that there is a real benefit outside of COVID to using these. Works in busy clinics, clinics that perhaps we wouldn't have been able to get any space in before. We can now get people to sign up at home. And also it works really well for those healthy volunteers who perhaps aren't attending the hospital. So we're going to continue to use that. But the other thing we're starting to look at now is, as I said, we've opened a COVID bioresource. So this was based on the same principle as the main bioresource. People who had been affected by COVID could sign up. Their blood would be taken and sent down to Milton Keynes, which was coincidentally where all the COVID testing for the country was happening anyway. And then all that cohort of people could be used for studies such as the ones you've seen, like Chris Duncan's the clinical trial study. These people could be invited to those because you had a whole pool of people who you knew had been affected by COVID. And we're now opening a branch of this called the COVID CNS study, which is to do with the clinical nervous system. So these are people who've had neurological complications that have been associated with COVID infection. And we're just in the process of opening that up now. But we're not quite there yet, but it's coming in the next couple of weeks. So this is just to let you know that that's coming. If you know anyone that's been affected by that, you can get in touch. So I think it's just a short talk today, just to introduce the bioresource and to talk about the kind of things that we do. But if you do want more information, we do have a website, but we also have a community on Twitter and on Facebook that you can visit. So I would encourage you to visit the website, the Biomedical Research website. We have a section on there, but also at Bioresource NCL is our handle on Facebook and on Twitter. So we do tend to post on there about the kind of things that we do research-wise and the kind of studies that people can get involved in. So I'll leave it there today so that we don't run over. And if there are any questions, hopefully we'll get a few answered. Thank you.
Thanks, Emma. Really interesting talk. So just one question that's come in so far in terms of, is there an age limit on signing up to the bioresource? Have you got sort of a minimum age and a maximum age, or is it sort of open to everybody?
Yeah, theoretically, it certainly was open to everybody. There was no age limit on the bioresource. In fact, I think our oldest participant is 94 and our youngest was about 13. But we have another new bioresource coming. It seems that 7 isn't enough bioresources for us. And we're going to be soon opening a children's bioresource. So at the minute, we're not recruiting anyone under the age of 16 because it's going to be more appropriate for them to be joining the children's bioresource, which is going to be aimed much more at them, and the kind of samples that they can give are going to be different. We probably won't take blood from them. We'll probably take saliva from them because of the safety issues. So yeah, so that's what at the minute, although we do have the ethical approval to take children's samples, we don't. But the rare disease bioresource, which as you can imagine focuses on there are 50 different rare diseases that we currently look at. That one often do take children because the rare diseases do span across all ages.
Thank you. And I think that's it from a question perspective on the bioresource. So I'll hand back over to Charlie.
Well, okay, that was great. Thank you so much for that, Emma. That was a really brilliant introduction into the bioresource. And that brilliant talk really now brings us to a close. So thank you all so much for coming along today and for getting involved and for asking questions and for really getting stuck in to learn more about the clinical research that's happening in Newcastle. I know that I've really enjoyed today. I've really gotten a lot out of all of the different talks. Just before we go, I do just want to let you know about a couple of opportunities. If you really enjoyed today, if you want to come along to more events, if you want to get more involved with research that's happening in Newcastle, there's some great opportunities currently active that you can get involved in. So if you're not already, you can join Voice. Voice is the online experience, the online network of members of the public that contribute their insights to ageing innovation and health research. And this was started at Newcastle University, but is now expanded nationally and globally as well. It's a really brilliant network of people to get involved in for research. There's also the NIHR Be Part of Research opportunity that you can sign up to register your interest in volunteering in research. Which is another brilliant opportunity to really start to get involved with what's going on in the world of health research. Finally, if you really are interested in the Newcastle Clinical Research Facility, there is a current opportunity now to join the Public and Patient Involvement Group to support the CRF and the brilliant research that they're doing that you've been introduced to today. We will send information about all these opportunities to you so you can take your first step, if it is your first step, in getting involved in research or maybe explore something new. We'll also send you an evaluation form, and it would be really great for us if you could just fill it out, just to tell us what you thought about today. If you thought it was terrible, which I'm hoping you didn't, do tell us though, because it's good for us to know, it's good for us to improve for future events. Did you like Zoom webinar or do you prefer the other Zoom? It's good to tell us this so we can make these events better for you and to let us know what you thought about today. But other than that, I just want to say a massive, massive thank you and a virtual round of applause to all of our speakers today for all of those brilliant talks. I think it was great to see such a breadth of variety of research that's going on in Newcastle. Massive thank you to Dominic as well for looking after those questions and managing that throughout the day, and to everyone else who's helped organise this event today to mark International Clinical Trials Day. But other than that, I just want to say again, thank you to all of you for coming along today, for getting engaged and getting involved with today's event. And hopefully we'll see you at another one very soon. Other than that, thank you very much, everyone, and we'll see you later. Goodbye.
And thank you, Charlie. Goodbye, everybody. Thank you.
Bye, everyone.